which is the loss of an electron. And so we know now that NADH really doesn't want those electrons. Okay, it just makes it less stable. I'll go over that again. And so it gives up those electrons. So if it's losing electrons, it's being oxidized. Each protein here at one point has gained the electrons, it's reduced. But remember, the protein right next to it is even more electrophilic, and so it loses it again, right? So this one is oxidized, this one's reduced. In other words, oxidation reduction reactions are always coupled. If one of them lost, then who gained? Okay, there's always an oxidizer, there's always a reduction, okay? So you want to keep it thinking about that. By doing this electron transport chain, as the electrons are taken away from NADH, I don't know if I can really draw on here. Okay, so I would say I prefer to have two little dots between the D and the H, indicating that, yeah, that's how the hydrogen is held on with a bond, but a bond is 2 electrons. And it's the 2 electrons that are gone, but what about the H? Well, the H minus electrons is a hydrogen ion, and that gets pumped up here at each and every passage of the electron. I told you I drew this myself, and this arc should have gone all the way across, so I apologize for that. Okay? But we have here a hydrogen ion gradient up here, which can't just flow right through because these hydrogen ions, even though they're really small, are charged. And the lipid tails of the plasma membrane repel. They just won't let it across. So they do come across, but through a very specialized protein known as ATP synthase. ATP synthase takes ADP plus a phosphate, and that's not shown on this picture, so please fix your picture, and it crimps it together to make ATP. Because this is an enzyme, it really likes ADP and a phosphate and has no interest in ATP, so it can keep making it over and over again. As the hydrogen ions flow through, this is what's happening. That's how the ATP is made. It's very successful in this, very efficient. It makes like 34 ATPs, and add that to the 4 you made before, you're looking at 38 total. Now, remember that oxygen, final electron acceptor. And we already established that oxygen, it's, an electron hog. Yes, we know this. So it's not waiting politely for its turn at this electron. It's stealing it wherever it can. And when it does so, now it's O2 with a dot. That's superoxide. That's highly reactive. It must be squelched. What does the arrow use to do that? Superoxide dismutase. Okay, superoxide dismutase. That's right. So this is what goes on, and this is why this is used. Okay, that's the role of superoxide dismutase in the arrow, is to tamp that thing down. Okay? But suppose you don't own superoxide dismutase, then you really shouldn't use oxygen, because now it's going to be superoxide and you won't be able to control it. So they use something else. They use nitrate, sulfate, and remember I said CO2, okay, to make methane would be the other thing, and I do have another slide that indicates that. All 3 of these, whether it's nitrate, if you think about where nitrogen is, it's just like 1 over just to the left of oxygen, and sulfate. Sulfur is below oxygen, so anything in the same column on the periodic table has the same characteristics, just a little less so. Yes? I'm not saying anything you don't know. Okay. So these are, and certainly CO2, those are not that electrophilic. They want the electron, but not the way oxygen does. So molecular oxygen is really pulling on these electrons, and that causes a bigger hydrogen ion gradient than what you're going to get with nitrate, sulfate, or carbon dioxide. And so this is why the arrow has so much energy, and they use that energy to divide. You're always going to see a ton of them up at the top of a tube. But when you're using something else, okay, you're making ATP, you will divide, but you're not making that much ATP, because the final electron acceptor is not as electrophilic. There's a smaller hydrogen ion gradient to flow through ATP synthase, so it's more like a trickle instead of like rushing water, which is what it would be like for molecular oxygen. That's just an analogy. Water is not rushing through ATP synthase. We're clear on that, yes? Okay.
Are SOD and catalytes coming into play
All over the place.
between the oxygen and water, or?
Oh, all over the place. This is what I'm saying. Like right here, at this point, NADH doesn't want those electrons and will give it to anybody at all. Okay, including oxygen. Right there. Could happen there. Could happen at the next step, this step, even the last step. Like it'll happen anywhere because molecular oxygen is an electron hog. It's trying to grab as much as possible. So I want to say two more things about this exact same image. Okay, so you can't really, no, can't lower it. All right, so because I would like to draw on it, but I'm not really going to be able to because I'll have to be on the wall above. So if you would for yourself, okay. All right, so if you would for yourself, you can put an X on one of these, okay? And that's what cyanide does. You've heard of cyanide, okay? It's a poison, it'll kill you. Okay, so we all know that, but like how? How does it kill you? Well, it stops one of the proteins in the electron transport chain. If we stop the passage of electrons, then we're not making hydrogen ion gradient. And if I don't have hydrogen ion gradient, what can I make? ATP. And so what happens to a cell without ATP? That's right. Why? It has no energy to do anything at all. Run the pumps to stay in the game, right? Remember that? I mean, we kind of like set that aside, but that's an ongoing process for every single cell. So not having energy will kill it. We know that, but like what? What's going to happen, right? Sorry?
Would that cause the cell to dehydrate and replace? Because
if it dies, if it just stops?
Yeah.
Well, yes, it would eventually like fill with water.
Okay.
Fill with water.
So it wouldn't be able to create the cell wall
Yeah.
or anything.
Yeah, it just, that will be the end of the cell. The cell absolutely needs to have ATP at all times. And as I told you, even for ourselves, if you did absolutely nothing, and I mean, these are from tables and stuff, I don't know that this is really true, but yeah, you know, if you weigh 150 pounds, then you need 2,000 calories a day to just run the system. Not even anything, like change the channel, that takes 2,000 calories, right? So if you're saying, I'm also gonna go to the gym and run and try to run after the 3-year-old, you know, who I'm babysitting today, like you need more, right? So there's, like just to run the system takes a fair amount of ATP, so cyanide is really dangerous. But if you would imagine right here, another protein altogether, okay? Just so instead of putting an X on one of these, draw like the same thing, but over here like another protein altogether. And that protein is the uncoupling protein, UCP. Uncoupling protein, UCP. What UCP does is it allows the hydrogen ions to flow through it, through UCP, instead of ATP synthase. So what happens then? Well, instead of converting the energy of glucose into the energy of ATP, and remember, you're only about 40% efficient at this, 60% is lost as what? Does anyone know? Heat. So the uncoupling protein, by instead of letting the hydrogen ions go through ATP synthase, it goes through the uncoupling protein. Now, all you're making is heat. That's it, only heat. This is found in two places, newborns and brown adipose tissue. This is done on purpose to maintain body temperature for that newborn. So you have to put yourself into the thought process of, you know, the newborn baby. What's going on here? You were just in a very warm environment that you did not create nor have to fight for. the way we're fighting this temperature here. Okay, we don't think of it as a fight, but you are in fact expending energy to maintain 37 against the 25 which you're in right now. Okay, for a newborn baby, they can't do that so easily. So they have an uncoupling protein such that they will break down their stored glycogen, because even babies have that in their, differing amounts, and how much did it weigh, et cetera, mom's diet. I mean, there's all kinds of stuff in there, but there's stored glycogen, which the baby will break down, and instead of converting that into ATP, converts it into heat. Okay, so it's breaking down its own stored stuff. It is very typical for the child to be born 7 pounds. Let's just throw something out. And then, bless you. A week later, you go to the pediatrician. They weigh your baby. Your baby is now 6 pounds 9 ounces. This is very typical. This is exactly what's going on. They grow up and that stops happening, but that is the way. And it's the uncoupling protein is expressed in newborns and brown adipose tissue, which is the other place where we can quickly generate heat if necessary. Does that make sense? Okay. Yes.
Is it true that babies I think I've learned this, that like babies have a higher temperature, like
Yes.
internal temperature?
Mhm.
Is that due to the uncoupling protein?
Their entire metabolism is running really fast. Yeah. So, and as we age, your metabolism runs really slow. I bet your grandma's always cold.
I'm always cold.
Right, I mean, it's just, right, and that has to do with other things as well, but it's just, metabolic rates are different. We all have the same metabolism, we're all aerobes, but the rate at which you're doing any process at all has to do with age and hormones and overall health and something with, blah, 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 but we're all into one. All right, so we're good here? Okay.
So are the uncoupling proteins only present in newborns? Like
And brown adipose tissue.
Okay, that's it though.
That's it.
Okay.
And the reason being, like, I really need that ATP. Okay, so, alright. So that's important to understand, and then recognizing that all cells are gonna utilize enzymes to do all their reactions, because it goes a lot faster, and I told you if I had to actually draw this, that this would be like way up there, right? Without the enzyme and with the enzyme lowers the energy of activation, that's what it's called, by orienting the substrate and putting them in the correct orientation, but also stabilizing that last little oomph. Okay, so with this, I told you that what this would be more like a multiple choice, like what's true or what's not true kind of thing, not for you to like spit it out. Okay, so just if you familiarize yourself with this. And then we went on to say that molecules have energy in their bonds. The bigger the molecule, the more energy in it. Okay, that's how that goes. And that the breakdown of a molecule like carbon dioxide, like glucose into 6 carbon dioxides is really to pop off all of these bonds. And we know that those bonds are electrons, and that gets captured to make ATP. Okay, so that's what happens as we go down. So from high energy to lower energy. And so the transfer of electrons, that's oxidation and reduction. We just want to know who's who and what's what. And super easy if you just remember mind and mind. Okay? Leo says, I mean, if oil ring's working for you, then you stick with that. That's fine. Okay, so our electron and hydrogen ion shuttle is NAD+. So I described this for you last time. And so it's a bigger molecule, okay, than what is drawn here, but this is the business end, just the upper part right here, okay? So here what you're seeing is that the difference between NADH and NAD+ is that NAD+ has double bonds around the ring, and we said that sometimes the double bonds are here, but sometimes it's, the other 3. And we said that the ability to share those double bonds all around is called resonance, and that's much more stable for that ring than this situation over here. You see this carbon there? This carbon has just one hydrogen, and that's fine, and then it can only ever make 4 bonds, so 2 to stay in the ring here, or sorry, 1 and 1 to stay in the ring, and then it can sometimes have the double bond here or here, no problem, right? Okay, so 1, 2, 3, 4. But as soon as we add an extra hydrogen, now this carbon can only ever make 4 bonds. So it's stuck. 2 to stay in the ring, 2 to the hydrogens, that's it. So it can't share these electrons. Now these 2 end carbons have to hold the electrons at all times. Electrons, remember, are energy. So now you're putting all the energy onto the 2 opposite sides, and that's less stable. This molecule would love to get back to this. All it needs to do is find something or somebody, I mean, I anthropomorphize, I'm sorry, some molecule to take away the hydrogen and the 2 electrons that hold it. Okay, so it is a reluctant electron and hydrogen ion carrier. This is where the whole thing begins. The electron transport chain and all that business, it's right here. Okay. Then we noted that, okay, being in the first 2, that's us, that's every animal, okay, fine. Lots of bacteria this way too. This is an excellent way to make ATP, but that's not the only way, okay? You can use something other than molecular oxygen, nitrate, sulfate, carbon dioxide. You're just not gonna make as much ATP. You see that it's depicted as such, like not that many cells compared to what you see for the aerobe. A facultative anaerobe has a leg up on an obligate aerobe in that, in the case of low oxygen conditions, that will kill the obligate aerobe, but the facultative anaerobe will instead ferment. Okay? If you're aerotolerant anaerobe, you're always going to ferment. Okay? And then these microaerophiles, they're kind of tricky to work with because they can't take it on the surface of a plate, too much oxygen. And they certainly can't take 0 either. So it's hard for us to, in room 317, to simulate those conditions. So we'll talk about this as we move forward, but we started the unknown today, and you may well have a mixture, right? Because everybody has 2, gram-positive and gram-negative mixed together. We're gonna find out. Okay, are you an obligate aerobe? Then that means that if I put you in a condition where there's no oxygen, you'll die. Are you a facultative anaerobe in conditions with no oxygen? I will see the end products of your fermentation, which are always acids and alcohols. I can check for that. Okay, so these are the tests that we're gonna do of who is this? Okay, because what we're gonna find out is like, okay, you're a gram-negative bacillus. I don't know who you are. I don't know what you are. Let's ask a few questions, okay? So, that. All right, so this was the summary of aerobic respiration, okay? We realized that we can eat something other than just glucose, yes, and things will come in at different places, but you see this? See this amine group here? Okay, that's the waste product of amino acids, of breaking down amino acids. Every single amino acid has an amine group on it. I mean, that's like definition. And that amine group, that's like ammonia, right? That's basic. So I can test for that too. So this is how lab goes, is like all these tests to figure out what's going on, okay? So the appearance or disappearance of a metabolic product, that's what we're doing. Okay, and then we just have to kind of recognize, we're just trying to find who's who. Okay, I'm not trying to turn everybody into microbiologists, okay? Just, this is what we use it for, because bacteria look alike, okay? All right, so here's just a picture of carbon dioxide finally as a final electron acceptor. But we said that that's not the only thing that can happen. Some organisms will not run an electron transport chain. They don't run a Krebs cycle either. They take the pyruvate, and what they do is that they peel the electrons and the hydrogen off of NADH, and they slap it on something organic. We defined organic to be 2 or more carbons. So it could be slapped on to some acid. It could be slapped on to some kind of alcohol. Those will be the end products. Okay, the end products of aerobic respiration we know are water and CO2. The end products of using nitrate is nitrite. The end product of sulfate is hydrogen sulfide gas. The end product of CO2 is methane. Those are all different, aren't they? Right, those are 4 different end products that I could test for, and for fermentation, we know that there's a lot of different end products. I ask that you know one organism and one organic end product. You're welcome to learn CO2 and hydrogen gas if you want to. That's not fulfilled the requirement. I still need to hear about something else, acetic acid, butanediol, whatever you choose, whatever will stick in your head, that's the one you want to stick to, okay? I think I took lactic acid off the table, didn't I? Did I take ethanol off the table? Okay, very good. All right, so lots and lots of choices there. And this is what we're doing. We're not quite there yet, but this is actually round 2 of the coliform analysis will be this. Okay, so it'll be designed to look for particular kind of fermentation and change in color. That's what we're going to do here on mannitol salt agar. This is going to also show us a particular kind of fermentation. So this is what lab micro is. You know, we have spent our time just trying to learn technique and how do you look for things, how do you inoculate, how do you plate, and now we launch. I mean, and it's like week whatever, but this is how it goes. We're in the intro class. From here on out, it's all like, which one is it? And we can figure this out by doing these tests. It requires, of course, being able to plate, being able to write like all the things. So we are not wasting our time. All right, is this where we ended up?
Yeah.
Okay, very good. So here's just a couple pictures of what's happening with the alcohol fermentation with yeast or lactobacillus and streptococcus like thermophilus that can make lactic acid, excuse me. And that is that with glycolysis, both of them make pyruvate, and 2 of them, okay? Because glucose is a 6-carbon molecule, pyruvate is a 3-carbon molecule, so you actually have 2 of those. And then notice here, for alcoholic fermentation, that one of the CO2s is dropped. You see that? Okay. If I had something called a Durham tube, so afternoon group you're about to find out, morning group you know what I just said, I could actually capture that as a bubble in a test tube. Okay. This will leave me with a 2-carbon molecule that goes through one more step to become ethanol. But what the real purpose of fermentation is, this is the purpose of fermentation, is to peel off those electrons and the hydrogen ion off of NADH to become NAD+. And the reason is I can't keep breaking down glucose unless I have NAD+ to add the hydrogens and electrons to. So that's the entire 100% purpose of fermentation, is to recover NAD+. Okay, so if you do make beer or wine, you have to allow for the release of CO2. That's part of it. If you make yogurt, pyruvate is a 3-carbon molecule as we described. So is lactic acid. So no CO2 release. Okay. All 3 carbons are still present in the final product. You see that? Okay, so yogurt's not frothy. If your yogurt is frothy, there's someone else in there. Okay, that's not lactobacillus and that is not streptococcus. I don't know what happened in the manufacturing process, or maybe it was you at home, something, but I know that that's not the case. Okay, lactic acid is a 3-carbon molecule, shouldn't have any gas release at all. Yes?
So, does fermentation always start with the pyruvate?
Yes,
Okay.
always.
And so, can you reiterate the purpose of fermentation?
Yes, is to regenerate NAD+.
Regenerate.
Because you're making NADHs in glycolysis, okay? So right here, like in glycolysis, you make NAD to NADH, right? And now I want it back. So NADH is kind of like, well, who's gonna take the electron and hydrogen, right? Like just somebody, something, take it, please.
Well, always produce two of them.
Two left.
Two NADs.
Yes. Yeah, we did not study the 10 steps of glycolysis, so that you would see at which steps they were generated at. Okay. So, if we just look at, okay, what's our final ATP totals here? You're really not gonna beat aerobic respiration. 38 for the prokaryotes. Why do they make more than us? Well, because there's this thing called FADH2, which I didn't even really spend any time on. That also will yield electrons. We're not great at that. We're not as good at it, but prokaryotes are. So they'll get every last drop out of a molecule of glucose. We're not bad, right? Two less. Anaerobic respiration. So look at this. It's fewer than 38, so not as good as aerobic metabolism, but more than 2. Why can't I give you a straight number? Because which one are you talking about? And there's more than this. Okay, the point is, it's not as efficient. Okay, so you have dropped in efficiency because your final electron acceptor is less electrophilic than molecular oxygen. Okay, fermentation 2, that's it. Not much. And look at this, how do you make your ATP? Substrate level. Okay, so oxidative phosphorylation is the electron transport chain, which does exist in both aerobic and anaerobic respiration. Okay, glycolysis is substrate level, Krebs cycle is substrate level, fermentation is substrate level. Oxidative phosphorylation is a specific thing, the electron transport. Okay, all right. So these are the different ATP producing mechanisms. This is the list of metabolic types. I ask this a lot. What is the metabolic type of this organism? I'm asking you, which tube is it in? It's hard for the student because you're just not used to it. Everybody's in tube 1. What do you mean metabolic type? Like, this isn't about you. Okay? There's lots of different kinds of metabolic types. All right? So, and we do know that the ability to tolerate molecular oxygen requires the detox reaction. So superoxide dismutase will do this reaction for you, and really, it'd be nicer if this arrow were much longer so that I could write the word superoxide dismutase over it, okay, so that you would know that that's what's doing it. But notice, what is my end product here? Hydrogen peroxide. That's also an oxidizer. That also steals electrons. That's got to be tamped down too. And for that, I have a second enzyme, catalase. Okay? So here's a few specific bacteria that we have described for each one of the different metabolic types. So obligate aerobes, this doesn't surprise you. Mycobacterium tuberculosis, where do you get tuberculosis? In your lung. That's like the most oxygenated place I have aside from my skin, right, which is exposed, right? Facultative anaerobes. So this is an organism that prefers an oxygenated environment, but it can in fact survive in low oxygen concentration. E. coli, other enterics, we're going to learn about those guys too, even Staphylococcus. But I showed you an actual picture of Staphylococcus versus E. coli, and E. coli is much better at it. Okay? Staph is not an organism that is typically found in the lower intestine, but we'll see when we get to normal microbiota that for some people it is, and it causes them some issues. Okay? E. coli is way better in the lower intestine, that's great. But so obligate anaerobes, all the Clostridium. So that's Titani, Botulinum, Perfringens, C. diff, all of those guys. Okay, obligate anaerobes. There's different bacteroides. You might have heard of some of these if you start looking into like what's normal microbiota in the gut. Those bacteroides. Okay, aerotolerant anaerobes. These are only going to ever ferment lactobacillus, streptococcus. Okay, so Streptococcus thermophilus, for example, is one of them, but so too is streptococcipyogenes, which is the causative agent of strep growth. It's an aerotolerant anaerobe. Aerotolerant anaerobes lack catalase. You see that? They don't have it. Okay, well, I should just memorize that. No, not really, but here's the deal. Are you somebody that you get strep throat every once in a while? You can kind of tell when it's coming on? Guess what? Here's a really low-tech solution for you. Take your hydrogen peroxide, dilute it, like 1 to 10 at least, and see if you can't gargle right in that spot where you feel it. It'll kill them. They can't stop hydrogen peroxide. Okay? Does that work on a staph infection? No, staph is a facultative anaerobe. It has SOD and catalase. That's like useless to you. That won't work, right? So you just kind of have to know who's who, what's what, and what can I use against them. There you go. Okay? All right, microaerophiles, Helicobacter pylori, but I told you like Neisseria gonorrhea is another one. Okay, yes.
Are microaerophiles only doing aerobic respiration with
Yes.
small amounts of, they don't do like fermentation or anaerobic?
They're just doing everything at like this little reduced rate, which is, you know, what they want, but they can't go to 0. That's the thing that'll kill them. So they're, it's like they can only live in really specific places. So Helicobacter pylori is not something that you're gonna get on your like upper lip. Like that's never gonna happen. It can't take that. Nor is it gonna be, even though it lives in the stomach, if it manages to dislodge and get swallowed and go to the lower intestine, it's going to die. There's not enough oxygen there. Yes?
This one causes the stomach ulcers, right?
Well, you know, I am in violation of COX 1 right off the bat, right? Because COX 1 says that I will see this organism in all cases of disease and never in the healthy. And yet Helicobacter pylori is like 50% of the population. So I don't think this is the cause. Right? They point at it. This is what happens. It's like just if there's a car crash and you happen to be standing on the corner, they're gonna blame you, but it's doing my own thing over here. Right? So, but you know, we're just gonna eradicate it. It's like, there's so much more to ulcers, which I can't even go into because I don't know, because you know why? Because it's not infectious. It's something else.
Does Helicobacter pylori produce any endotoxins? Or produce
No, it's a gram negative.
Okay.
I mean, it has an endotoxin, because that's lipid A. That's just part and parcel of being a gram negative. But you need a lot of lipid A to cause the problems that come from endotoxin. All right, so now let's move on to the